there appeared to be an increase in cytotoxicity on the addition of TNF-alpha. So at that point is when I started my Tempo-Seq experiment. So I took these chemicals along at the lower concentrations with the co-stimulation with not just TNF-alpha, but also interferon gamma. But when I did this in triplicates, this was after the Tempo-Seq was done. While the same trend was there, there was not much of a significant those cytokine co-stimulation effect. However, I had already done my Tempo-Seq by then. So my hypothesis was that the technique can be used to explain mechanism of toxicity, can be used to predict toxicity using biomarkers that can be seen in the data at low doses, and it can be used to explain drug cytokine synergism for ideally at low doses. So these were my samples for Tempo-Seq. The first three here were the entry chemicals for which I only tested in the chemical itself, no co-stimulation. But for the ideally chemicals, I had unstimulated TNF-alpha and interferon gamma together, and 3 concentration of each, while here I had 4 concentrations. So this is how I did the analysis of Tempo-Seq dataset. So this was done with my colleague Jonathan, who has more experience with informatics. So first I started with a row-count matrix from BioSpider and did the weighted gene co-expression network analysis. Then I did the normalization and DSEC2, which is for obtaining the differentially expressed genes. And WGCNA is to cluster the data set entirely into different modules based on co-expression. And this can be further enriched for the GO terms for determining which functional pathways they fall into. And then this overlap will tell me which pathways or where the differentially expressed genes fall functionally. And then there's a correlation with the dose response. So here in the volcano plots, you can see the 5 chemicals for which I had differentially expressed genes for the concentration model. And as expected, since olanzapine and gentamicin, which I took along, but they did not have any dose response. So in fact, gentamicin was one of my controls. So for them, I did not have any differentially expressed genes. For diclofenac, actually, the plot is a lot bigger than this. I had quite a few more differentially expressed genes, but I'm representing the top ones because otherwise the plot is a mess. Then I did a WGCNA analysis and identified the modules that had genes that were co-expressed. And the width of the module and the color denotes which module it is in. The width is representative of how big the module is in terms of number of genes. So then the Go enrichment was used for classifying the modules as per the functions. So these are the colors written here, and for each color, this is the name that I gave based on the functional terms. So this is RNA metabolism, UPR, ER stress, xenobiotic metabolism, ECM, interferon signaling, channels and transporters, ribosomes, protein processing, mitochondrial processes, respiration, cell cycle of 2 types, and 2 parts of the cell cycle, and lipid metabolism. So these were, after the WGCNA analysis, the sizes of each module. And in each of these modules, this is the relative size in terms of the number of differentially expressed genes in each. So cell cycle 1 and 2 and UPR and xenobiotic metabolism happen to have the most differentially expressed genes. However, this goes away in the 72-hour time point because probably because of a washout and because the doses are not that high. And also the maximum differentially expressed genes were observed in amiodarone and diclofenac, as I showed before in the volcano plots. Here I represent also the additive effect of TNF-alpha, the additive effect of TNF and interferon gamma, and synergism or antagonism, as the case may be. So in these 4 chemicals, I had the co-stimulation. So only over there you see the other bars. but it appears to have only an additive effect, and only when there is TNF and interferon together. And it appears to be mostly an effect in the interferon signaling. So I don't think there is, and since there is no synergism that I see, or not that many, this method does, the Tempo-Seq does not seem to be predicting, ideally at least at this dose. So maybe at a higher concentration it can. So there, in the genes in the UPR pathway and mitochondrial processes, the modules correlate very highly to dose response at these concentrations tested. And on the x-axis, you have the module membership, which denotes the cluster centrality. So how central or how important a gene is to a cluster. So the ones on the right are the most correlated with dose response. So in conclusion, I have the modules of UPR, ER stress, xenobiotic metabolism, and mitochondrial oxidation found to be most altered in response to chemical treatment, and this is well known from existing literature. And the genes involved in development of toxicity were observed in the results as well, so such as ICAM1, ATF4, ATF5, HSPA5, and 6 were observed in amiodarone and diclofenac, and even in ibuprofen, and this is also known. INSIG1 is involved in the development of obesity in response to treatment of clozapine, and this also popped up. And interestingly, also S100P, which is a known biomarker for acute kidney toxicity caused by ibuprofen, was also differentially expressed in the data for ibuprofen treatment. However, the method seems to be inadequate for drug cytokine synergism prediction or explaining the mechanism. The only significant synergism that I had was with diclofenac in these three genes. But there is some evidence of ALAS1 involvement in hepatotoxicity downstream of drugs like rifampicin and isoniazid, which are also ideally drugs. And this is mostly, this is probably due to the pathway of heme production. So there is an accumulation of protoporphyrin, which is toxic for the cells. So ongoing and future work, I'm also doing the determination of timeline of expression changes in the key genes involved in toxicity development, and use of ICAM1 and HSPA5 reporter lines to detect the changes in expression induced by chemical treatment. And ICAM1 and HSPA5 are both genes that came up, which is why I'm using them. And thirdly, I have the benchmarking of my data against available PHH datasets for the same chemicals to compare the ability of the model to detect genes involved in toxicity development. So they're all ongoing. Lastly, I would like to thank my PI, Catherine. All of them are my team members, or both current and previous. And whoever I've not mentioned here are also very, I mean, I thank them too for all their help and guidance. And definitely all of the M3 team of PIs and students for being there with me throughout this journey, and for all their help and guidance. Thank you.